Watch this little green sphere stretch, pinch in at the middle, and split cleanly into two. It is dividing, the way a living cell does. But nothing inside it was ever alive. It was assembled, piece by piece, from a shopping list of purified chemicals. This is a synthetic cell, a cell-like bubble built up from the outside in using only ingredients a chemist can name and measure out. A fatty membrane for the outer shell, a stripped-down machine for reading genes and making proteins, and a genome of its own, written by the team. It comes from Kate Adamala and Aaron Engelhart's labs at the University of Minnesota, posted in a July 2026 preprint by Gout, Deitch, Cash, Hoog, Engelhart, and Adamala. To see why that is hard, compare it to the usual way people make a minimal cell. That approach starts with a real bacterium and deletes genes until barely anything is left that still lives. You end up with something simpler than nature, but you never left nature. This project runs in the opposite direction. Every part started as a purified molecule in a tube. The membrane, the couple dozen enzymes that transcribe and translate genes, the building blocks, and the DNA were all combined on the bench until the mixture started behaving like a cell. The reason this counts as a milestone is what those parts do together. A living cell does not just sit there. It takes in resources, grows, copies its genome, and splits into two that carry that genome forward. String those steps into a loop, and you have a cell cycle. This synthetic cell runs the whole loop. Let me walk through each step, because each one had to be solved separately and then linked. Start with feeding. The cell cannot make everything it needs, so the team hands it groceries in the form of other, simpler bubbles called feeder-liposomes. These feeders are packed with fresh enzymes, building blocks, and extra membrane, but they carry no DNA. The trick is getting them to merge with the cell instead of just bumping into it. The cell's surface carries a tagged protein, and the feeder's membrane carries a matching lipid that grabs that tag. When they lock together, the two bubbles fuse, and the cell swells with new material and more membrane to grow into. Here is a clever part. How do you prove the cell actually fed, and fed again, generation after generation? Each feeder carries a short strand of DNA that acts like a tally mark. When a feeder fuses in, it drops off its tally strand. Count the strands, and you can tell how many rounds of feeding a cell and its descendants have been through. That turns an invisible process into something you can read off in the lab. Next, copying the genome. For this, the cell uses a workhorse enzyme called Phi29, borrowed from a virus that infects bacteria. Phi29 latches onto a circle of DNA and reels off copy after copy in a rolling motion, like thread unspooling. To be sure the copies were genuinely new and not just the original DNA they started with, the team chemically marked and then destroyed the parent strands and checked what survived. Fresh DNA showed up only when Phi29 was present. They tracked several separate pieces of the genome this way, including the gene for the green glow you saw at the start. Then division. Real cells often pull themselves apart using an internal skeleton of protein filaments. This cell has none of that. Instead, the team first split the cells the blunt way, by pushing them through a fine filter that pinches large bubbles into smaller ones. That gave them reliable division to run their experiments. Then they showed a version the genome itself can drive. Crowd enough of that tagged surface protein onto the membrane, and the strain forces the bubble to constrict and break into two. Division without a cytoskeleton, encoded in the DNA. Now, connect the loop back to the DNA, because that is where it stops being chemistry and starts being biology. Division here is tied to a gene the cell carries. So when a cell divides, its daughters inherit the same genetic recipe that let it divide. Change that recipe, and you change how well the offspring feed, grow, and split. That is the setup for the last step, and the most striking one: selection. The team made a second version of the cell with one change: a stronger switch driving that surface protein gene so it makes more of the protein. More protein means more fusion with feeders, faster growth, and more daughters. They mixed the stronger cells with the ordinary ones and let them compete over 5 generations. The stronger variant pulled ahead and took over more of the population, and its edge grew sharpest when food was scarce. A helpful trait written in DNA, spreading through a population by out-reproducing its rivals. Assembled from non-living parts. It is worth being precise about how far this goes, because the honesty is part of what makes it impressive. The complete cycle was demonstrated for 5 generations, and each generation takes about half a day under carefully held temperature and salt conditions. After those 5 generations, only about a third of the cells still carried the full genome. So inheritance works, but is still leaky. The cell cannot make its own enzymes, energy, or fresh membrane from scratch. It depends on those feeder liposomes and a controlled lab. And this is a preprint, not yet peer-reviewed. This is a cell you can build and drive, not a free-living organism.
So why does a fragile 5-generation bubble matter? Because building something is the hardest way to prove you understand it. For decades, researchers argued about the minimum a cell needs to run a life cycle. This work answers part of that by construction, and it lands under the size people expected. A genome of roughly 90,000 base pairs was enough. And a cell assembled from known, labeled parts is a chassis you can redesign on purpose. Swap a gene, tune a switch, and watch what changes, without a billion years of evolution in the way. The next milestones are clear too, closing more of the loop so the cell makes its own parts, and pushing past 5 generations. For now, the news is simpler. A cell cycle just ran in something that was never born.